Hello and welcome to the School of Success. My name is Roy and I'm very happy to have you here in our second video on viruses. In today's video, we are going to be discussing about the genetic materials of virus, and let's get into it. As we have discussed in the first video, viruses are very small particles, and that's a limit to how much things can be packed into a virus. In a broad sense, a virus is made up of two simple things, which is the genetic materials and also the capsid. In today's video, we are going to be focusing on the genetic materials themselves. At the mention of genetic materials, perhaps the double helix structure of DNA comes to mind. While some viruses do have double-stranded DNA like ours, many actually have different forms of genetic materials, and we are going to examine deeper into that. There are four main factors to examine when classifying genetic materials of viruses. We have prepared them into tables, so let's head over to the other screen.
Alright, the first factor that we will be discussing will be about single versus double strands. So in a single-stranded genetic material, it will look something like this, while a double-stranded will look something like that. The immediate difference that we can tell will be that the single-stranded genetic materials should be smaller in size than that of a double-stranded one. Next, in terms of stability, as the double-stranded genetic material has its complementary base pair, it will be more stable than that of a single-stranded one. And this will also mean that it is more likely for the single-stranded genetic material to mutate as compared to a double-stranded one. Do note that in terms of mutation, it occurs randomly, and with more mutation, it means that there will be more genetic variation present in the gene pool. Since mutation occurs randomly, it is possible for mutation to cause the virus to be less effective, and that's a good thing for us. So do not assume that mutation is always a scary thing that is going to make the virus into some super virus. Yeah. Moving on to RNA versus DNA. RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. It has something to do with the ribose sugar that makes up the genetic material. And one of the other obvious difference that we can notice will be the base itself. Ribonucleic acids have a base of uracil, while deoxyribonucleic acid has a base of thymine, as you may be able to notice over here. Once again, DNA is more stable than RNA itself. RNA has a higher tendency to be degraded by the host because RNA can result in the production of respective proteins, so the host will have some regulating mechanisms when it comes to RNA as compared to DNA. Hence, RNA itself is not only less stable, but is easier to be detected and also destroyed by the host. With that being said, since RNA can be used to produce proteins immediately, the steps to process RNA will be significantly lesser than that of DNA genetic materials, as DNA needs to first be transcribed into RNA before it can be used to produce proteins. Moving on to our third factor, which is whether it is circular, linear, or segmented genetic materials. A circular genetic material may look something like that. It is comparatively easier to form because there's no need to create caps at the end of the genetic materials. There are studies that claim that this also prevents the shortening during replication, because during replication itself, some form of the genetic materials may be lost at the end of the genetic sequence. Next up, we have the linear form of genetic materials, which can look something like that. In order to protect the genetic materials from being degraded at the ends, they will require caps to be formed at the end. And while this may require more processing to produce the caps and place them on the ends of the genetic materials, these caps can serve other functions like promoting the processing of the sequence. Last but not least, we may have segmented genetic materials, means that there may be more than one piece of genetic materials found in a viron, which is a unit of the virus. Usually, it consists of a few linear segments, hence this makes the virus itself more complex, and since the virus is more complex, usually it will take more processes to produce the next batch of viruses. However, at the same time, with more segments in the genetic material, it is possible for the virus to carry out different processes at the same time because, they are not dependent to one another and the processes can just roll on. And finally, to our fourth factor, whether it is a sense, antisense, or ambisense genetic material. Sense, we commonly represent it as a plus, antisense as a minus, while ambisense will be a plus/a minus. What does the sense strand means? The sense strand usually refers to the RNA where it can be used immediately to produce functional proteins, while the antisense strand must first be transcribed into another RNA before it can be used to produce functional proteins. Last but not least, ambisense means that that strand of genetic material will have a sense and an antisense strand embedded within it. So what are the advantages and what are the differences? First and foremost, like what we have described, a sense strand, since it can be used immediately, it will require less processing as compared to the antisense strand. And for ambisense strand, because it has both, it usually requires more complex mechanisms to be able to ensure that both of those processes can be carried out correctly. And in terms of complexity, definitely the sense strand will be less complex. Now that we have gone through all four factors which we will examine when it comes to viral genetic materials, let's take a look at some common examples. So over here we have the influenza A virus, the virus that's responsible for causing measles and mumps. Our coronavirus, which is responsible for SARS, MERS, and the current COVID-19. The virus responsible for chickenpox and shingles, as well as the one that's responsible for herpes. Take a look at this table and you may realize that, hey, we have some that are in the single-stranded RNA, while another, which is in the double-stranded RNA. To account for this difference, it is because of the difference in strategy that the virus are using. Both single-stranded genetic materials and RNA are less stable and are more likely to mutate. And this allows for more variations to be present in the gene pool and hence allow the virus to have a higher chance of survival if the environments were to change. As for the double-stranded DNA approach, we can recall that double-stranded genetic materials as well as DNA are more stable and has a lower rate of mutation. This allows the virus to have a more consistent approach because their genetic materials are not changing as much. And also in most cases, these viruses usually have an elaborated defensive strategy which is already tested through time.
While viruses are not considered living things by many scientists, due to the presence of genetic materials, we can actually use these sequences to group them into different families and groups and eventually produce a taxonomy of viruses. This is particularly useful because it can help us find similarities and differences between viruses. So what's the significance of this knowledge? By knowing the genetic makeup of viruses, it allows us to create better taxonomy of viruses. And this will greatly reduce the area of research that scientists have to conduct to find out more about viruses, especially when it is a race against time, like the current battle against SARS-CoV-2. In fact, at the discovery of the novel coronavirus, the first thing that scientists do was to sequence the genome of this novel coronavirus. And in that process, they found great similarity with the virus that has caused SARS in 2003. This finding greatly reduced the amount of effort and time required for scientists to discover what type of proteins can be found on the virus itself. And in fact, there are already vaccines against COVID-19 that is ready for trial. We will be discussing more about antigenic proteins in our next video. And if you're interested to find out more about antigens, please check out our very first video on this channel. Do you know that mapping of the viral genome in patients can also help with contact tracing? As viruses are prone to mutation, it is possible to trace back the connections and sequence of infection by comparing the similarity and differences between the viral genome in patients. So over here, we have prepared a simple scenario to illustrate this. Take for example, if patient number 4 and patient number 5 were to be diagnosed at the same time and both of them live in the same estate. It will be easy to assume that both of them have contracted the virus from the same source. However, through the analysis of the viral genome, it was found that that of patient number 4 is highly similar to patient number 2 and patient number 3, who happens to be his parents. The viral genome of patient number 2 and patient number 3 are highly similar to patient number 1, who is the first infected to be isolated. This allows a confident claim that the tracing between this group of patients are complete. However, it is discovered that the viral genome of patient number 5 is distinctly different from that of 4, 3, 2, and 1. This will then inform us that there is some untraced infected who has yet to be surfaced in the community. And the relevant authorities can then step up in terms of measure in limiting community activities. It turns out to be yet another very long video, and thank you so much for sticking through once again. If you have any question, feel free to comment down below, or you can also head to our website where we have a Q&A bulletin and you can post your questions there. If you find this video helpful and you'd like to see more of these contents, give us a like and subscribe to our channel. Remember to hit the bell so that you will be notified when we post our video, most probably weekly. And yeah, we will see you in the next video. Cheers!